Hello everyone. I am Kondo Park, and I'm excited to share our sleep foundation model, called SleepMaMi. This work is a collaboration between the College of Medicine and the Graduate School of Data Science at Seoul National University. This research is about sleep. Sleep is an essential part of our lives. Everyone sleeps, no matter your age, or whether you're rich or poor. But rapid urbanization has changed our lifestyles a lot, and most people today don't get enough sleep. Because of this, more and more people are paying attention to sleep quality. At the same time, the number of people with sleep problems keeps growing. According to Korea's Health Insurance Review and Assessment Service, the number of patients diagnosed with sleep disorders is growing by about 6.4% every year, and more than 1.3 million people were diagnosed with sleep disorders in 2024 in South Korea. The best way to understand sleep is to analyze polysomnography or PSG data. PSG is the standard test used to study sleep. During an overnight PSG, many different physiological signals are recorded. This gives us rich information about sleep and can support many different clinical tasks. Researchers have built AI models to understand and analyze PSG data. But existing PSG-based AI models are usually built for just one task, or just one or two types of modality. Because of this, they don't fully use all the rich information that PSG recordings contain. On top of that, most past research has relied on supervised learning. But the labels attached to PSG data are made by human scorers, so they aren't fully objective. For example, inter-rater variability exists in PSG scoring. The agreement between different human scorers is only around 80%. And because clinical scoring guidelines get updated over time, the correct label for the same data can change depending on when it was scored. Another key limitation: most existing sleep AI models focus only on sleep microstructure, the short-term patterns found within individual 30-second segments. But sleep also has a macrostructure, the overall pattern of sleep across the whole night. Understanding both micro and macro structure is important for a complete picture of sleep. To solve these problems, we built SleepMaMi, a PSG-based sleep foundation model that combines both macro and micro structure. The model has two main parts: a microencoder, which learns fine-grained physiological patterns, and a macroencoder, which captures sleep dynamics across the full night. The model was pre-trained in a self-supervised way, using a combination of masked autoencoding, contrastive learning, and a new method we call demographic-guided contrastive learning. It was trained on more than 20,000 PSG recordings, about 158,000 hours of sleep data in total. Let's look at each part in detail, starting with the microencoder. Its goal is to learn a universal representation and modality-specific representation across different PSG signal types. To do this, we use private and shared encoders. Private encoders capture information specific to one signal type, while shared encoders learn information common across all signal types. We then use contrastive learning to align these representations into one unified space. The shared features from different signal types form a positive pair. We use two types of negative pairs. The first is a representation negative pair. The private and shared features from the same signal type are pushed apart. The second is a temporal negative pair. Two shared features taken from different points in time are also pushed apart. On top of contrastive learning, we also use a masked autoencoder. During this pre-training, the model learns to reconstruct parts of the signal that were hidden or masked. This helps the model learn fine-grained features for each type of biosignal. The second main component is the macroencoder. Instead of looking at each 30-second epoch on its own, the macroencoder processes the entire sequence of sleep epochs across the whole night. We use a Mamba architecture, which can efficiently model these very long sequences. We run Mamba in both directions, forward and backward, so the model learns the sleep pattern from both the beginning and the end of the night. To pre-train the macroencoder, we introduce a new method called Demographic Guided Contrastive Learning, or DGCL. Think about visiting your grandmother's house. When you wake up, she's already been awake for hours, doing all sorts of things. You've probably noticed that her sleep pattern looks very different from yours. This isn't just a casual observation. Differences in overnight sleep patterns across demographic groups have been well studied in sleep medicine, and our own data confirms it too. The chart shows sleep stage distributions for two different groups, and you can clearly see how their full night sleep patterns differ. Inspired by this, we use contrastive learning to build the macro structure latent space, guided by three factors: age, sex, and BMI. We chose these three factors because they're free from scorer bias, they don't change when clinical guidelines are updated, and they're included in almost every dataset, which makes this approach easy to scale to larger datasets. Let me walk through DGCL with a simple example, using 3 subjects. First, each subject's full night sleep features are split into fixed-length cycles, and we compare subjects cycle by cycle. Then, subjects who are demographically similar are pulled closer together in the latent space, while subjects who are different are pushed apart. Here's the learning objective, written as an equation. This generalizes standard contrastive learning. Instead of a simple same or different label, we use a weighted similarity score between each pair of subjects. That weighted similarity comes from the demographic distance between two subjects, based on how different their age, BMI, and sex are. For pre-training, we combine 9 public sleep datasets, giving us a large-scale pre-training set of more than 20,000 PSG recordings from a diverse range of people. We tested SleepMaMi on several downstream tasks. First, for sleep staging, SleepMaMi outperforms every baseline we compared it to, including other time series and physiology-focused foundation models. For sleep-disordered breathing segmentation, SleepMaMi again consistently outperforms other foundation models. This task requires a fine-grained understanding of sleep microstructure, and SleepMaMi does well here thanks to the microencoder's MAE and contrastive learning pre-training. For disease prediction, SleepMaMi performs on par with the current state-of-the-art, showing that it can also capture subject-level patterns, not just short-term signal patterns. SleepMaMi also shows strong few-shot performance, staying accurate even with very little labeled data. This tells us the representations it learns generalize well across different tasks and datasets. We also confirm the real benefit of the macroencoder directly, across sleep staging, age and sex prediction, sleep apnea-hypopnea index prediction, and major disease prediction. Adding the macroencoder improves results over using the microencoder alone. To conclude, SleepMaMi is a new sleep foundation model that jointly models both sleep micro and macro structure. It's pre-trained in a self-supervised way on more than 20,000 PSG recordings. We combine masked autoencoding and contrastive learning for the microencoder, and introduce DGCL, a new method, for the macroencoder. SleepMaMi achieves strong performance across a wide range of sleep-related tasks, and shows excellent adaptability. Thank you for your attention. Please stop by our poster or visit our website for more details.